Spermatogenesis. Introduction. Gametes are mature haploid germ cell or sex cells. Male gametes, that is sperm, or female germ cell, ovum, fuse with another mature haploid germ cell during fertilization to form a zygote. Gametogenesis is the formation and development of cells that form gametes or sex cells in male, that is sperms, or females, that is oocytes, in the male and female gonads from precursor cells. Gametogenesis in males are called as spermatogenesis. The process in which undifferentiated germ cells or immature or primordial germ cell undergo differentiation to form mature sperm. Gametogenesis in females are called as oogenesis. This is the development of female sex cell or oogonia into mature ovum in the female gonad, that is ovary. Formation of the primordial germ cells. Formation of the primordial germ cells. During the early embryogenesis, the 5th week of gestation, the primordial germ cells originate in the endodermal epithelium of yolk sac in the vicinity of allantoic stalk. These cells migrate to the gonadal ridges, which later give rise to functional gametes in males and female gonads, testis and ovary respectively. These cells remain dormant until puberty, and at puberty, these cells differentiate to form mature sperms or ovum. This process continues throughout the life but decreases at old age. Normal anatomy. The testis is a paired ovoid male reproductive organ situated in the scrotum. From the superior aspect, it is suspended by spermatic cord. The epididymis is a comma-shaped body situated on the lateral part of posterior border of testis. It consists of head, body, and tail. The head of epididymis receives and stores sperms released from the seminiferous tubules of testis. The sperms in the epididymis undergo maturation and travel through the tail of epididymis into the vas deferens. The testis is enveloped by a double-layered membrane known as tunica vaginalis. Except at the superior and posterior border, where the spermatic cord and epididymis adheres to the testis. The testis is composed of convoluted seminiferous tubules in which the process of spermatogenesis takes place. It is held in a loose connective tissue with numerous elastic fibers and interspersed Leydig cells. These Leydig cells often present in groups. These cells are the prime source of testosterone, that is male sex hormone. The seminiferous tubules consist of a basement membrane on which the primordial germ cells lie. These germ cells differentiate or mature as they advance towards the lumen of the seminiferous tubule. Also interspersed between the spermatogonia are the Sertoli cells, which are large pyramidal-shaped cells with centrally placed nucleus and nucleoli with numerous cytoplasmic organelles such as Golgi complexes, ribosomes, lysosomes. These cells project inward from the basement to the lumen and provide support, nutrition to the developing sperm cells, secrete fluid into the lumen and are phagocytic in nature. The spermatogonia and the spermatids are embedded in the recesses of the Sertoli cells throughout its development. Spermatogenesis. The first stage of spermatogenesis occurs at puberty, where the primordial germ cells differentiate to form type A dark spermatogonia, produce more type A dark and type A light spermatogonia. The type A dark spermatogonia have the ability to self-renew, and the type A light spermatogonia cells differentiate and undergo mitotic division to form type B spermatogonia. These type B cells further divide to form primary spermatocytes. The primary spermatocytes consist of 46 chromosomes, of which 44 are autosomes and 2 are sex chromosomes, that is, X and Y chromosomes. These diploid primary spermatocytes enter first meiotic division and produce haploid secondary spermatocytes, that is, 23 chromosomes in each spermatocyte. These secondary spermatocytes then undergo second meiotic division to form spermatids, which possess 22X and 22Y chromosomes. These spermatids gradually undergo a series of morphological changes to develop into a mature spermatozoa. This process is known as spermogenesis. In this process, each spermatid comprises of a spherical nucleus, Golgi apparatus, centrosome with proximal and distal centrioles, mitochondria, and cytoplasm. The nucleus condenses, and the Golgi apparatus with acrosomal granules coalesce and spread over the anterior pole of the nucleus to form a bilaminar cap that covers over 2/3 of the nucleus. The centrosome occupies the posterior pole of the nucleus and splits the proximal and distal centrioles to give rise to axial filaments of the body, that is, middle part, and the tail of the spermatozoon. The posterior centriole forms the ring centriole. Around the axial filaments in the middle part, the mitochondria aggregates and forms a mitochondrial sheath in a helical manner. From the ring centriole, the axial fibrils develop and extend posteriorly into the cytoplasm of the cell as it becomes elongated progressively. As the spermatids mature, most of the cytoplasm is extruded and it detaches from the cell, thereby becoming the residual bodies. These residual bodies are engulfed by Sertoli cells. The mature spermatozoa frees itself from the Sertoli cells and enter the lumen of the seminiferous tubule. This process is known as spermiation. The entire process for one primordial germ cell to form mature sperms takes 64 days. These spermatozoa are non-motile and are transported passively from the seminiferous tubules to the epididymis, where they are stored till they obtain full motility. Structure of spermatozoa. Mature spermatozoon is a specialized and actively motile cell. It is composed of head, neck, middle piece, and tail or principal piece. The head has an ovoid shape that measures about 4 microns in length. It contains a nucleus with 23 chromosomes, that is, 22X or 22Y chromosomes. The anterior two-thirds of the head is covered by a bilaminar acrosomal cap. This cap contains enzymes such as protease, acrosomase, acid phosphatase, and hyaluronidase, which helps in the penetration of the oocyte during fertilization. The head is enveloped by cell membrane and is devoid of cytoplasm. The neck is a small constriction between the head and middle piece. At the posterior pole of nucleus, two centrioles are seen, that is proximal and distal centriole. From the distal centriole, outer fibrils of the tail develops. The neck is enveloped by cell membrane that is continuous with middle piece and tail, and it contains small amount of cytoplasm. The middle piece is long cylindrical in shape, measuring about 7 microns long and 1 micron in diameter. This part consists of axial filaments surrounded by mitochondrial sheath, which are enveloped by cytoplasm and cell membrane. The axial filaments consist of a central pair of microtubules within a symmetrical set of 9 doublet microtubules and an outer ring of 9 coarser fibers. A ring centriole is present at the junction of middle piece and tail. The tail or principal piece is the motile part of the cell. It is the longest part that measures about 40 microns long and 0.5 microns in diameter. It contains from within outwards axial filaments, fibrous sheath, thin cytoplasm, and cell membrane.